So my name is Isaac Galatzer-Levy. My background is in clinical psychology and computational methods, machine learning, and artificial intelligence. I primarily got to know this group of researchers through my work at NYU, where I've been involved in computational models around PTSD, really specifically around how we can harness the knowledge we already have to build real-world predictions of stress responses, how we can build those into emergency medicine or military contexts, really in line with what Eric and Yossi are talking about in terms of a golden hour, where we understand a lot about the biological development of post-traumatic stress. And a real focus of my research has been on how can we harness that information to make real pragmatic decisions about who's at risk and what our treatment needs. I currently am the chief scientific officer at a company called iCure, which is an artificial intelligence company that focuses on developing app-based softwares to measure facial and vocal characteristics to flag clinical risk. And I'll talk a little bit about that work because I think it's also very relevant. And I'm going to try to ground everything a little bit in the conversation I've started with Eric and Yossi and Irina, really around, how could you build out methods to spot risk or intervene in very complicated situations like those that NATO deals with, where you have diverse populations, you have diverse languages, diverse cultures. How can technology really be of service to that? So everything I'm going to talk about, I'm going to try to ground that a little bit in the really pragmatic goal of how can we really help people in the real world at scale. So these are my disclosures. I am an employee of iCure, and as a startup, I get stock options. I also do a little bit of consulting. None of it, I think, is that relevant to my presentation today. So I want to start with just some ground setting before we get into complicated models, which is the world's a dangerous place. I used to give this slide and people didn't really think of, the first world as so dangerous compared to sort of these images of some places that are at least not in my experience, but it's not really true. The majority of people are exposed to a life-threatening event at some point across their life course. So whether wherever you live in the world, roughly 80 to 90% of people will experience some sort of life-threatening event, which is qualifying for post-traumatic stress. What's really interesting about that is that we don't all respond to those kind of events the same way. So we go in very different directions. And a real focus of my research, but I think a lot of people's research on this panel is how come? Why are people going in different directions? Why are some people adapting while other people are not? If we could really understand that on a mechanistic level, we can harness that information and it can be very powerful. We're used to seeing, really the very negative effects of trauma. These are images of people who've experienced some very traumatic events. At the top right, these are children being led out of a school after a mass shooting, which is a common event, unfortunately, in the United States. But people are also use these events to be quite resilient, come together. We see that right now across the world, the ways that these difficult events can bring us together and help us grow. The top right, these are kids returning to Sandy Hook Elementary just a few weeks after that mass shooting. So, and human beings, like all species, we experience, trauma and difficulty in our lives, and we try to continue on and carry on, and we're not that different from other species that way. So the prevalence of PTSD, despite the high exposure, is actually surprisingly low, and that's something that, we've been debating now for at least as long as my career. How come? We have 89% exposure, but we only have about 8 to 15% psychopathology long-term as a result. So we can really model this, we can understand how people respond in terms of a few different kind of prototypical patterns of response. So if we look at a severity index, like say PTSD severity or even depression or other sort of outcomes of trauma, and we look at time, we now know that there's really 3 common patterns of response. There's people who are able to carry on, who don't develop much of a stress response. people who develop a severe stress response but are capable of recovery, and people who develop a real chronic stress response. So this is obviously an oversimplification, but if we think about these, breaking down these prototypes, then we can start thinking about what influences these outcomes. Why do people end up on these trajectories? And really consistent with what Yossi's talking about, the golden hour, how can I know that really at this beginning point here when I could potentially affect those trajectories? So, you know, we can call this chronic recovery resilient. We can call them apples, oranges, and bananas if we like. But what we know is that people are very different in their response in some, you know, not random ways, some meaningful ways. So really the first person to really work on this that I know of was my PhD mentor, George Bonanno, and he identified these different trajectories of response to loss and trauma. And he found that people really do tend to follow these different patterns, and that the majority of people tend to be resilient. We've now replicated that in a lot of different contexts. So these are trajectories of both depression and anxiety in people who had a spinal cord injury that caused para or quadriplegia, which is very, you know, stressful. These are military personnel who were deployed to Iraq and Afghanistan. This is US soldiers. Here we have the trajectory of response to a heart attack in large population samples. And this is police officers. This is work I did with my postdoc mentor, Charlie Marmer at NYU, showing that police officers who are chronically exposed really also follow similar trajectories of response, and that they're really predictable before they're actually deployed into police service. And finally, this trajectory is spinal cord. This is another spinal cord injury sample from a large population. We see these same sort of response patterns. Now, one thing I did early in my career that I found very interesting is to try to model those same types of response in some of the animal paradigms we use to study PTSD. So these are actually trajectories of what we call fear conditioning and fear extinction, where I couple a shock with a tone, and I look at how does an animal respond to the tone once I take away the shock. And what we see is three common patterns once again in terms of extinction learning. So we have animals where if I pair a tone with a shock, they'll start freezing more and more, as we see on the y-axis all the way to the left. And then when I play the tone without the shock at repeated measures, the majority of animals extinguish very quickly. So they stop freezing in response to the tone, and they learn, okay, I'm in a new context where the tone no longer means I'm about to receive a shock. We have about 30% who that takes more time. They follow the slow extinction trajectory, and roughly 10% that fail to extinguish. So no matter how many times I play this tone without a shock, they just keep on freezing up. So this really indicated to me, at least, that there's a real biological basis to these patterns of learning and memory that influence post-traumatic stress response. And we can start to really look translationally at what influences these trajectories. So if we look back at these images I showed earlier of the horrors of trauma, we can actually look at them in a more biological way. So we see this boy here who's clearly very dissociated. We see the amount of upset and arousal on people's faces. And we now know a lot about the biological underpinnings and actually the role that arousal or that response plays in the biological adaptation to stress, or potentially the development of post-traumatic stress through mechanisms that influence learning and memory, and a lot of the things that Emily Holmes studies. So I won't go too deep into that. But we know that the pathways to post-traumatic stress outcomes are really quite complex and involve a lot of different systems on both sides of the blood-brain barrier. So this is a schematic that is, you know, purposely a bit confusing, but this really represents the different pathways between different hormones like epinephrine, norepinephrine, CRH, cortisol, how they interact with other features like beta amyloids and tau, cytokines, and influence across the blood-brain barrier and influence different neurodevelopment in different regions of the brain, or cell death in different regions of the brain. So trauma has these really robust effects across the blood-brain barrier, both in our central and peripheral nervous system. These are all the systems that Yossi and Eric have really defined over their careers, and I've just capitalized on you know, what they've been able to put together. There's so much happening but it's not random. And if we can understand how this system works and when it works in relation to trauma, then we can harness that information to make real world predictions or real decisions. And that's really the goal for my work, is how can I capitalize on the deep science that's been developed over the last 30 years in this area, both in animal models and human research, to make real pragmatic decisions. So what does all of this stuff do to us? Well, it actually affects a lot of systems. It affects circadian rhythms, physiological response and regulation, long-term potentiation, which affects memory formation, brain aging, glucose metabolism, immune functioning. So all of these are activated in response to stress and ultimately influence how the event affects us. And it does it by changing some clinical characteristics like sleep-wake cycles, arousal, memory, and cognitive functioning. And these pathways really are not unique to PTSD. They affect have affected a number of different disorders like PTSD, MDD, but also the same pathways affect the development of Alzheimer's disease, influence schizophrenia outcomes. So we really actually are thinking about, when we study PTSD and stress responses, we actually really have a great window into really a transdiagnostic picture of how people adapt to stress in their environment, either in a resilient way or through the development of psychopathology. So from my view, PTSD is actually a really great methodology to study how the brain responds to stress generally and how psychopathology generally develops. PTSD is really the only disorder we have where we know the genesis point. As opposed to schizophrenia, we don't really know. Same thing with Alzheimer's. So we have an opportunity to actually understand the development and then understand how to influence it, which makes it very exciting. So by doing that, by really separating out different populations, we can start to look at the mechanisms that influence them along these sort of complex pathways and then make some real decisions. So here, this is actually fear extinction trajectories in human beings, and we find the same types of trajectories of response when we measure eye blink startle. I've made sure, Eric told me that there are lots of residents and students, so I've made sure to include all of the papers. And I'm happy to share my slides to be of help. We find that genes like FKBP5 that regulate the cortisol binding in regions of the brain along those pathways really influences those trajectories in human beings and really differentiates particularly hyperarousal symptomatology. So we know that as we start to break down these constructs, we can better understand the underlying biology, and then we can translate back and forth between humans and animals. So what you're looking at here is actually, once again, trajectories of extinction learning in animals, this time in mice. And what we did is we gave a high dose of dexamethasone, which we know is going to affect that stress responsive system. And we looked at the expression of that same gene, FKBP5, in the amygdala. And so the amygdala is very important in the development of PTSD. It's very well characterized how memories are formed in the amygdala and the connections between the amygdala, hippocampus, prefrontal cortex, and other regions of the brain. And what we find is that if we give a high dose of dexamethasone after the animals have acquired fear before they start this extinction training, we can actually experimentally manipulate the animals to all follow this rapid extinction trajectory. So I give a high dose of dexamethasone in that period after they've learned this relationship between a shock and a tone, and I can actually rescue the animal and I can help him to prevent him from being, or her, from being in this non-extinction trajectory. So this is really a nice way to think about exactly what Yossi was mentioning about this golden hour. Is there a time where those memories are developing, similar to Emily Holmes' work, where is there a period where I can really manipulate the stress response to get an optimal outcome? So that's really what we can see on an animal level, and we want to know, well, first, can we spot who among that 100% of trauma-exposed people actually will be that 10 to 20% who will develop PTSD? Can we understand those same mechanisms, and can we manipulate them in the right period? So if we really want to identify people at risk, what we can do is we can use methods emerging from the machine learning literature, where we can say, okay, I have these different populations, chronic stress, recovery, resilience, and I have a lot of information about what influences that response. I know their immune system, their metabolic response, their arousal response are all going to influence that outcome. But they relate to each other in a very, very complicated way. So I can never really disentangle that complexity. What I can do is I can take those indicators and put them together and say, well, can they really tell me this person is at risk? So what I'm going to do is say, okay, I have these three populations and I have a lot of these indicators. Can I build a model that says you're going to be on a chronic trajectory, resilient, or recovery? And if I build that model, what can I then validate it in an independent sample and show that it really works? Eric Vermetten and I wrote about this concept, and we tried it with clinical data some time ago. The article, it was in Journal of Psychiatric Research. And we tried to really build this out. So I had a grant from NIH to do a study at Bellevue's ER. Bellevue is a great place to study trauma because we get roughly 30,000 admissions a month. It's very high flow emergency room. We recruited 190 people in the ER and we followed them for a year to try to identify their trajectory. And we collected an enormous amount of data in the emergency room from electronic medical records, indicators of those responses. So there's actually a lot in white blood cells, in other types of just CLIA labs that tell us actually about the stress response. Even characteristics like blood pressure that are taken upon admission, we know are associated with the PTSD trajectory. Can we harness that information? And when we designed this study, we really designed it to have data that matched very closely to a much larger study, the Grady Trauma Project, run by Tanya Jovanovic and Carrie Ressler at Emory University, so that we could replicate our algorithms and across samples. So we took that data and we tried to build an algorithm to predict PTSD. So this is the prevalence of PTSD in the two independent samples, very similar. In terms of 12-month prevalence is 15% versus 22%. And we tried to use information in the emergency room from electronic medical records along with some stress items we collected at the same time to try to predict what trajectory will people follow over 1 year. And this is the area under the ROC curve for the model building sample, independent replication. And what we found is that we could predict PTSD trajectories at roughly an 86% accuracy just based on the stress response in the emergency room itself. And the markers that really drove it did replicate across samples as well. So we found that subjective indicators of stress were predictive and are important to include. We also found that number of white blood cells and markers of metabolic change were also highly predictive. So we published this just recently in Nature Medicine, where we really spelled this out. And what we encouraged was the inclusion of a number of stress items in electronic medical records that can then be seamlessly integrated to make a prediction. So this is really where we want to get to, and this is what Yossi and Eric brought me to this NATO group to talk about, is really, could you build actually a machine? Could you build an algorithm that takes in this information in an automated way and says, this person's at risk, and gives the information to the clinician so that the clinician can make a decision about their care? Can I spot that 10% so I'm not trying to intervene with 100% and wasting 90% of my resources? Now, the area that I'm very interested in is, how do I take this further? So I can spot a certain amount of risk, but now with the ubiquity of cell phones, I can actually continue to collect data after somebody has left the emergency room in really a scalable way that's very low cost. So there's a huge emerging area known as digital biomarkers. How can I use sensor data or cell phone data to really track somebody's response? And what we know from work from Eric Vermetten and others is that, while we know that if we can intervene very early in the emergency room, that's ideal, but we actually have a window of about a week or so to really spot people at risk, and we can change their trajectory by reaching out to them, offering them already empirically validated psychotherapies and treatments. So what would I collect? What would I measure if I can measure somebody over a cell phone? What are indicators that are of interest? I'm just going to point, and the answer is, there is no good answer to that question. I can tell you what I think. And what I think are the things that we have always thought are clinically relevant. How can we measure them objectively? So this is actually an interview with a patient with major depressive disorder I had to watch as a trainee. And when I was trained to learn how to identify how a patient is doing. This is a patient with depression, and even without the sound, you can see that she's depressed as a clinician. She's showing very prototypical signs of depression. Her eyes are lowered, her gaze is lowered. She's not showing eye contact. You can see the sadness on her face. Generally, she's showing little motor activity. She's fidgeting with her hands. You could hear her speech. The content would be sad. Her rate of speech is quite slow. So we've always treated those kind of characteristics, the sitting with a patient and talking to them, as subjective and not objective, but that's not really true. Just as the previous examples I pointed to, these markers that we see on people's face or in their voice really do represent the underlying biological functioning, and it's being communicated to us directly, and we understand those signals well. So what if we could actually build machines to measure those directly, and could we do it over a cell phone? So with the emergence of methods like computer vision and voice analysis, I can actually directly measure facial characteristics. So here's a nice neurological example. This is Muhammad Ali, who had Parkinson's, and what I'm doing is I'm measuring asymmetry on his face.
Can you use your mouse to indicate where you are on the slide?
Oh, yeah, let me see. How do I do that? I'm not sure how to, will this work?
We can, yeah, exactly. That's
okay.
right.
Yeah, I see. So here's Muhammad Ali when he's younger. I'm going to show a symptom where I'm literally going to measure one side of his face versus another to look at asymmetry, which is a symptom in Parkinson's disease. So let me see if I can. Oh, I think I have to turn off my cursor to click this. Sorry, I don't know how I did that. Okay, so here is just looking at the asymmetry between his mouth, eyebrow, and eye, and as he gets older and more Parkinsonian, he becomes more asymmetric. So this is just a simple example of a symptom that we know is relevant to the disease, but it's very hard for us to measure, as a clinician or observe clinically, but I can do it just very directly by measuring his facial activity. Here's eye blink, which we use to measure startle and arousal. But it would be very difficult for a clinician to say count how often somebody is blinking or their intensity. Here I'm able to do it very directly just using these computer vision methods. So and here's another example where we can actually measure arousal, actually pulse directly from somebody's face by extracting different layers of color and looking at their change. So here's an individual actually measuring pulse just remotely over a cell phone with video. So we can start to get at some of these arousal characteristics and mood characteristics directly and remotely, and also in a transcultural way, which is really important if we want to think about how do we build out systems that could really deploy in any context. So to test this idea, we went back to that Bellevue data where we did a qualitative interview with people at 1 month following trauma. We just sat them down and said, tell me what happened to you, and just made them talk for 4 minutes. What we did is we then extracted features, including facial characteristics of emotion like fear, anger, disgust, the intensity of their emotion. Also, we looked at their prosodic characteristics, like how fast were they talking. We also did natural language processing, so we could look at the valence of their speech, all in a totally automated way. And here are the results cross-sectionally. We were able to classify cross-sectionally whether people have PTSD or depression based on cutoffs at roughly a 90% accuracy. And in terms of an R squared, which is how much can we predict the severity based on those markers, we were able to account for roughly 60% of the variance. I'll share this paper with you so that you can really look at the markers. It was just published recently in Psychological Medicine, but at the end of the day, it was a combination of meaningful features like how much fear were they showing, how much anger were they showing, how also other characteristics like age, how they used language, that was really predictive of their clinical functioning. We did a similar type of study to see if we could passively measure suicide with Birgit Klein at University of Zurich. We sat people down after being admitted to the hospital following a suicide attempt, And we did a similar type of just qualitative interview, what brought you to the hospital? And we saw how vocal and facial markers predicted suicide severity scores on the BSS. And what we found were that markers of voice percent, so that's how slow people were talking, so the amount of white space literally between words is what we were counting. Facial expressivity and head movement all were predictive of how suicidal people were. So what we found specifically was that people who were more suicidal talked slower, showed less facial expressivity, and their heads moved significantly less. I thought that was very interesting because we understand now mechanistically that patients with who are suicidal have a very low serotonergic tone. Serotonin has a very strong effect on motor functioning. And really what we're observing here is really a low motor profile where people are talking slow, they're moving slow, and their facial characteristics are quite blunted. When we look at actually how do people respond to SSRIs, so this is some of my work at iCure, where we looked at the same markers in patients with major depressive disorder and said, how do they respond when we put them on SSRIs? We see those same markers respond to SSRI treatment. So in red, what you see across these three graphs is the MADRS score, or the depression score, and people are going from significantly depressed to below the clinical cutoff. They also significantly decrease in the amount of negative affect on their face. They significantly increase in the amount of overall affect. And their speech becomes much faster and their head moves more. So when I actually directly manipulate serotonin, what I find is that these characteristics actually are treatment responsive. So they're not trait characteristics, but in fact state characteristics. So long story short, I see these panel of measures that reflect underlying biological functioning being something that could be deployed in the real world to really measure clinical functioning in a transdiagnostic way, but also a transcultural way that is really language free. Which is really exciting because when I went to Odessa to meet with this group, this was the question that was posed: how could you do that? How could NATO do this in different regions with different languages, with different populations, really at scale? And when that question was asked, my answer originally was, you can't. It's not feasible. But I think if we look towards markers that are really transcultural and easy to deploy at a very low cost at huge scale, I think that's very achievable to really spot people at risk and understand what are the mechanisms that can be manipulated to improve their trajectory. So I want to thank everybody. I want to very much thank Eric and Yossi for inviting me. And I want to thank all of the collaborators who helped collect this data and analyze it and publish it. Thank you.
Thank you very much, Isaac. We can briefly unmute to applaud for your presentation, and then mute again. Thank you.
Yeah. Thank you very much, Isaac. Couple of points. One is, you know that I'm obsessed with the time of the day. Are you going to consider it as, you know, what time of the day? And the other point is regarding the cortisol. I didn't see that you look at cortisol level at the ER, but I might miss it.
So I'll answer both of those questions. So time of day, yeah, I mean, this is a perfect example. 5 years ago when even 5 years ago when we were talking and say, well, how can we even make sense of time of day? Somebody comes to the emergency room and, when did the trauma happen? When were they assessed? All these kind of characteristics. One of the beauties of technology is, once you start building automated systems, you have exact timestamps for information. And so now, at the end of the day, you can do something with information you have. You can't do anything if you're blind to it. And so we can start thinking about those labs in the context of the time of day, in the context of time from trauma, all in ways that we used to only be able to do through self-report, which just introduced an enormous amount of error and really couldn't solve our problem. On the question of cortisol, I think that's a very good example. So when we designed the study, we asked the question, what everybody's looking at in these predictive models are mechanisms that are very interesting but not accessible. So I was involved in a number of large DoD-funded studies for biomarkers of PTSD, where we looked at esoteric mechanisms like proteins or gene expression, and those are just very, very expensive. So if we're talking about real-world deployment, you know, we're not going to change the way that emergency medicine works. We're not going to start burdening. In the US, that would turn into a $100,000 bill for every patient who comes to the ER. We can't do that. So how do we think about something like cortisol that we know is mechanistically important? Well, look at eosinophils, which we already collect and have a very strong correlation and mechanistically directly relate to cortisol functioning. And I can just get it for free from labs that are already collected. So we want to think about what we in machine learning call information equivalence. Are there things that tell me about something that's really important but hard to get, like an fMRI signal? So could a cognitive test tell me about, you know, a region of the brain? You know, so I'm not going to understand stress in the center of Belarus, you know, by putting everyone in an fMRI, but I may be able to deploy a cognitive test, you know, on their phone. So for me, you know, understanding those mechanisms and then how they relate to proxy markers is really super important if we want to make it scalable.
Thank you. Let me ask you also a question, and this is not a nitty-gritty going into the individual parameters or so, but actually more perspectivistic or so, or more visionary. And let's zoom out a little bit, because you're targeting computational psychiatry. You didn't mention the word, but this digital phenotyping or so over a time course. Let's zoom out and take a perspective of where this field is going. You're introducing a novel compound with novel technology. In 5 years or so, if we think big or so, what is your perspective of where the field is going, and where you see that your expertise would be implementable or so into clinical practice? I know not a 10-minute or so, but could you give us a little bit of your insight and your idea?
Absolutely. So I'll answer that question as best I can based on conversation, you know, me and Eric actually had a couple days ago. So Eric, you know, very nicely wrote a commentary on my Nature paper, and he asked me, this is the first time we talked last week, and he said, well, what's the response to it? You know, and I said, well, you know, the truth is people like it, but there isn't the massive response, you know, one would hope for, because the truth is in the United States, for example, people don't necessarily want to know if somebody's getting sick, because it costs them money. So the question is, how are systems going to pick up these methods? How are militaries, how are hospital systems, how are payers, providers going to pick up these methods and move them forward? And that's always the case as a scientist. You know, you develop methods and you say, you know, this is the right answer and this is how to do it, and let me spell it out for you. And then how can you influence systems? So one of the reasons I like being in industry is you can start to influence those systems, but also it's made me realize that most of the players we have are not necessarily that motivated to, say, make people healthier always.
That depends, Isaac. Our next presenter will be Emily, and of course we want to know if we're infected or so with COVID. So it's just
Right.
a signal that we're all interested in to be picked up or so. I want to know if I'm bipolar and if I'm getting manic or so. So it depends on what you're looking at, whether the society at large will be interested in.
Right. So that's the key point is, you know, how do you actually make sense and implement these in the pragmatic problems that healthcare is trying to solve, you know, and that's how you move from science into the real world. So a good example is, you know, I did some work in, could you predict relapse in people with addiction? And insurance companies are very interested in that problem. Now, the truth is insurance companies don't care so much if you're addicted. They care that people who are addicted go to the emergency room a lot, and they're very expensive. So you have to find the ways in which you can overlap and see benefit. Same thing with military systems. You know, in the US, somebody develops PTSD and the Veteran Affairs, you know, is responsible for their care for the rest of their lives, which is very expensive, you know, and to be fair, people in the VA are very concerned with how to have good outcomes for our soldiers and our veterans. And so how do you find that overlap? And that gets beyond the science and really gets into the pragmatics of how do these healthcare systems operate and how can you really kind of get into them and influence change. So that's actually a lot tougher, I think, than the science. It's my take home.
Thank you. There's a remaining question from the panelists or so. Just, I didn't see them on the chat. If you have a question and you're in the audience or so, please use the chat function. But I do want to give the priority, if I may say so, to Joseph, David, Emily, or Irina. Not, or
Thanks, Isaac. It's really intriguing what you're doing. A lot of it's very new to me. But of course, we do know that one of the best predictors of bad response to trauma is previous trauma. And previous bad. I mean, how much can you explain your data just in terms of that previous history?
Yeah, I think that's a very good question because no question, prior trauma and early childhood trauma, robustly and always are predictive. We also know that there's measurement problems with how we account for childhood and previous trauma. We in effect ask through self-report, and we haven't really been able to figure out a good method, and especially in the kind of framework I'm thinking about, how do you do it in a very automated way? So I don't have a good answer, but I can tell you the experiments that I've done. So we actually recently did an analysis we're going to be pushing out where we used US Census data. So if you give me your zip code, I can get a lot of information about your neighborhood. In fact, I can get a lot of information about your neighborhood when you were a child, by asking the same thing. So we took census data and we actually factor analyzed it. And what we found is that there's clear characteristics like how many murders are there in your neighborhood, how many hospitals, how many schools. And we created a factor analysis of those census data. And we basically created 3 factors, which were really clear, which were how dangerous is your neighborhood, how well resourced is your neighborhood, and those are highly predictive actually of the PTSD trajectories. So I see that as actually an opportunity where, once again, literally from your medical records, I can get your zip code and I can pull that data and I can integrate it into my models. Now, other countries don't have a zip code system, but they have other systems and census information. So I see that as an accessible way within this framework to just be pulling in that information.